Hi everyone, I'm Ambika, a graduate student at Penn State, and today I'm really excited to share with you some of my work on positive and negative chemotaxis of enzyme-coated liposomes. The objective of this work is to try and understand how can we steer colloids or protocells in both positive and negative chemotactic directions. In particular, today I'm going to examine and rule out several existing transport mechanisms for these soft colloids and show you a potentially new and previously unrecognized mechanism of transport based on solute phospholipid interactions. So, why are we so interested in directional migration? The ability to sense chemical signals in its surrounding environment and move accordingly is crucial for the survival of living systems. To give you a few examples, directional migration enables cells to go towards food, away from toxins, and coordinate collective behavior such as tissue formation and biofilm formation. Here is an example of a vesicle that is being transported across a cytoplasm of a cell through the conversion of chemical energy into mechanical motion. Here is another example of a white blood cell that is able to sense a foreign particle in its environment and move towards it, finally engulfing it. This kind of motion is called chemotaxis and is generally seen in two directions. It is called positive chemotaxis when the cell migrates up the gradient of concentration or towards higher concentration. It is called negative chemotaxis when the cell migrates down the chemical gradient or away from high concentration. Inspired by nature and biology, scientists all over the world have fabricated many synthetic material systems that is able to display chemotaxis based on chemical and biochemical reactions. Here in the four examples that I show you, whether it's enzyme molecules, enzyme-coated hard particles, or catalase encapsulated within soft particles, they show positive chemotaxis. In the enzymatic synthetic systems, negative migration is less studied and has not really been demonstrated before. More recently, soft motors such as lipid vesicles or polymerisomes have come under the spotlight due to the various advantages that they present over inorganic hard particle motors. So firstly, they're biocompatible and they resemble the membranes of cells. So this makes it easier to for them to be used as drug delivery motors. And also they can be used as artificial cell models for a variety of studies ranging from origin of life or how virus particles would attach onto the cell membranes. In this work, what we wanted to see is in what direction enzyme-coated lipid vesicles would move when they're placed in a gradient of reactant. So similar to the previous studies where positive chemotaxis was seen, we also hypothesized that when enzyme is coated on the external surface of lipid vesicles, we would see positive chemotaxis. In order to test this hypothesis, we employ mainly three techniques. The first technique is thin film hydration for lipid vesicle synthesis. After thin film hydration, we extrude the lipid vesicles to a uniform shape and size of about 100 nanometers. The lipids that are used to make these liposomes are shown here. The first lipid has a rhodamine molecule that enables us to visualize it under the confocal fluorescence microscope. The second lipid has a biotin head, which allows us to attach enzyme onto its external surface. The third lipid and the backbone lipid constitutes the backbone of the lipid vesicles, which is about 95% of lipid vesicles. After we make these uniformly sized liposomes, we want to conjugate enzyme onto its external surface. We do this through the enzyme biotin streptavidin linkage and coat them on the external surface of these extruded, uniformly sized vesicles. So once we form these active vesicles or enzyme-coated liposomes, we want to test their chemotaxis in imposed gradients. And we do this by using the 3-inlet, 1-outlet microfluidic channel. So typically, we flow the lipid vesicles through the mid-channel And the side channel has buffer, and this constitutes the control experiment. When we replace one of the side channels with substrate or reactant, this is the experiment. And we compare the experiment with the control to obtain the amount of movement that we get in relation to the substrate. So here is a typical graph that we obtain from this experiment. The blue curve is the control, which shows the movement of the active liposomes into the side channels through Fickian diffusion. When we replace one of the side channels with a reactant or substrate, we see there is some amount of movement, and we call this as the chemotaxis, chemotactic movement of the lipid vesicles. So the first enzyme that we wanted to test was catalase. So catalase in literature, we have seen that enzyme catalase molecules as well as catalase-coated hard particles show positive chemotaxis. Similarly, when we attach enzymes onto the external surface of lipid vesicles, we also see positive chemotaxis. So there was no surprise there. The next enzyme that we tested was urease. Now, urease molecules as well as urease-coated hard particles show positive chemotaxis. So it was so surprising to us that when we attached urease onto the external surface of lipid vesicles, we actually observed negative chemotaxis. Now, why were we seeing this negative chemotaxis? We thought that there would be some interaction of the lipid vesicles with the solute that caused this negative motion. So first, what we did was we removed the enzyme from the external surface of the lipid vesicle and tested just inactive lipid vesicles in gradients of urea, the reactant. But we don't see much movement. That led us to believe that it is the product of the urease reaction that is causing this negative chemotaxis. And that is exactly what we tested next. We tested inactive lipid vesicles in gradients of ammonium carbonate, and indeed we see a negative chemotaxis. Not only did we observe this for ammonium carbonate, we saw that there are a number of solutes that cause lipid vesicles to move in the negative chemotactic direction. So this led us to allude or hypothesize that there is an additional mechanism of movement acting on soft liposome particles. So that brings me to the next part of my talk. What exactly is the mechanism of negative chemotaxis in lipid vesicles? For the remainder of the talk, I'm going to discuss inactive liposomes moving in gradients of solute. So we had a couple of hypotheses that is listed here, but spoiler alert, we were able to rule them out, and our current standing hypothesis for lipid vesicle movement is according to the Hofmeister series. So let's look at the first mechanism first. So diffusiophoresis is the mechanism by which colloidal particles are able to migrate in gradients of electrolytes or non-electrolytes. For today, I'm talking to you about electrolyte gradients. So electrolytes means salts, and salts have anions and cations that could have difference in diffusivities. So according to the difference in diffusivities of the anion and cation, there could be a spontaneous electric field gradient that is created depending on which salt you use. So here we used two salts, ammonium carbonate and sodium chloride. Due to the difference in diffusivities between the anions and the cations, the electric field that is formed is opposite to each other. So to rule out diffusiophoresis, we tested the motion of lipid vesicles in the gradient of oppositely electric field causing salts. And while ammonium carbonate makes the lipid vesicles go in the negative chemotactic direction, we expected to see sodium chloride to move it in the reverse positive chemotactic direction, but we don't see this. So we can effectively rule out the diffusophoretic mechanism for colloidal liposomal transport. The next mechanism that we had in mind was density-driven flows. So in this case, what we did was we replaced the lipid vesicles in the mid-channel with solid particles, 200 nanometer polystyrene beads. So since polystyrene beads are heavier, if there were any density-driven flows, we would see them at the interface where the particles interact with the gradient of solute. However, we tested this and we do not see any movement, and so we can effectively rule out density-driven flows in our system. The next hypothesis that we had was osmophoresis. So osmophoresis is the movement of membranous colloids when placed in impermeable gradients. Here what happens is, due to the osmotic pressure difference inside and outside of the vesicle, the liposome expels water from the side facing high solute concentration and admits it on the other side, propelling the lipid vesicle away from the high solute concentration region. So in this case, what we did was we compared our experimental value with theoretical osmophoretic values. And we saw that we were able to get an overlap between the two only with a huge factor of 10 to the power 4. So we can say effectively we were able to rule out osmophoresis because osmophoretic transport in our system is negligible. So at this point, we had covered majority of the traditional mechanisms that's available for colloidal transport, and we were wondering what exactly is going on in our system. So we went back to literature and we found the Hofmeister series. Now, the Hofmeister series is a series of anions or cations according to their ability to salt in or salt out organic biomolecules. So this series has been around since the 1800s, but it has not been used to show transport of colloids. So in this series, if you go from the left to the right, you go from well-hydrated salts to less well-hydrated salts. So ions such as carbonate, ammonium have a good water structure, and they're able to aggregate proteins or biomolecules in solution. Whereas on the other side, they do not have a good water structure, and they're able to solubilize, increase the solubility of organic or biomolecules. So carbonate do not associate well with the hydrophobic part of organic biomolecules and repel them, while thiocyanate is able to associate with the hydrophobic part and attract them. So what we observed when we looked at the Hofmeister series is that the ions that actually cause negative chemotaxis of liposomes all lied on the left side of the Hofmeister series. So in order to check whether Hofmeister series is really causing the lipid vesicle movement, we picked a salt from the opposite end of the series to see whether we're able to get a reverse positive chemotactic effect. And that is exactly what we saw. In this case, the carbonate ions that are well hydrated are repelled or depleted from the lipid-water interface, while thiocyanate, which is less well hydrated, is enriched at the lipid-water interface. So according to the Hofmeister series, we saw that the depletion or enrichment of ion at the lipid-water interface causes positive or negative chemotaxis. And in summary, in this work, we were able to show that active and inactive liposomes move in both positive and negative chemotactic directions. We were able to effectively rule out diffusiophoretic, osmophoretic, and density-driven transport mechanisms. We also showed that our lipid vesicles are moving in accordance with the ions in the Hofmeister series. And also, this kind of movement is a first step in programming directional motion in adaptive life-like material. With that, I'd like to conclude my talk and thank all both my advisors, my collaborators, and my lab mates, and of course, Penn State and the funding source that made this research possible. I'd be happy to take any questions, and please feel free to ask me anything that you would like to. Thank you.